Good morning. My name is Zachary, and I'm a grad student here at UMBC, and I'm going to show you what it's like to be a graduate student here working in a microbiology lab.
I usually begin my day by walking through the quad between the Commons and the Recreation Center to get to the Biological Sciences building where the lab is. Once I get to my building, I take a brisk walk up the stairs to the lab. Once I'm in the lab, I quickly get settled in and begin working for the day. This always starts with putting on gloves and cleaning my bench. For my experiment today, I had some bacteria growing overnight in the shaking incubator. Let's see how they did. As we can see, it looks like they all grew well. I then head back to my bench and then the freezer to grab some reagents for the experiment I will be doing today, which is polymerase chain reaction, more commonly known as PCR. Once I have everything I need at my bench and my bench is set up, I can begin labeling and pipetting all the chemicals I need into each tube. These tubes are small, so I have to be really careful. Once I have everything I need in the tubes, I get to place them in this cool machine that makes a lot of copies of the DNA that I want for each of the cultures that I grew overnight. I then let it sit and run for almost 2 hours. While it runs, I get to focus on some schoolwork and updating my lab notebook. Before the reaction is over and the machine finishes, I have to go and set up the next portion of the experiment, which is making this cool jello-like gel that I can check to see which of the reactions work. I do this by microwaving the gel until it is super hot and bubbling. Hold tight, it might take some time for it to become fully liquid. Once it is all turned into a liquid, I can pour it into a tube, mix it with some other chemicals, and then pour it into a mold that makes it the right shape for me. I then wait some more for the gel to become solid and the other machine to finish. Once both the other steps are done, I take the tubes and add some blue dye to them. I then add all the reactions to different wells on the gel, and I can now watch them move down the gel due to electrical current. After about 30 minutes, I check on them and we can see that they have moved down the gel. Now it's time to see if some of my bacteria have the DNA I want. I then place the gel on this box that shines UV light on it and makes the DNA light up. As you can see, the first gel I made only had one set of DNA per lane. This isn't what I want. However, in the second gel, we have 2 bands in most of the different lanes. This means that they all worked. Now, I can tell which bacteria I want to keep and grow for other experiments in the lab. So I take the bacteria and send them on a ride in the centrifuge to separate them from the food that they have been growing on. Then I pour out the liquid and all I have left is the bacteria all clumped together at the bottom. I can then put them in the freezer for use another day.
Well, thanks for watching. If you have any questions regarding what I do in the lab or the Biology Outreach Initiative here at UMBC, please feel free to reach out to me or anybody in our group. Look forward to speaking to you soon. Thanks.